Hello biology students, this is Mr. Ekman and I'm putting together a video to help you guys review how to do the act of translation, how to go from an RNA molecule to an actual protein. How do we find what the amino acids are? And we use this diagram right here, what we call the universal genetic code, to make this happen. And this kind of started when we were working on this page, if you remember this yellow page where it says protein synthesis practice. And we were giving you some options here, right? We filled in some of the parts, and then as this went on, it got a little harder where there were other parts that weren't completely filled in. So taking a look at this, I want to review this with you here and just go through a couple of these again in case you need some help on this more than what we did in class. So let's look at this very first question because we're going to see this one more often. Our very first item started out here with our DNA strand was TAC. And we know this is DNA. We know DNA has got the base pairs G, C, A, and T. And we're going to go to an RNA molecule right there. We're changing DNA from RNA, and this is what we call transcription. And we'll change this to G, C, A, and U. Now we've got G cow bases, not G cat bases. So those are different. That's why we see all the U's here in RNA and the T's in DNA. So we want to make this jump. So T is going to pair up with A when we do transcription. A is going to pair up with U because now there's no T's. Thymine is gone, uracil is in. And C is going to pair up with G. Cytosine finds guanine. So that's how we end up with our 3 letters for RNA, AUG. Now that 3-letter combination is what we call a codon. It's the thing that genetic code is on, is a codon, the 3 letters of RNA. And we read the codons. We read the genetic code to figure out what amino acid we're gonna make. And this amino acid is already filled in for us. It's methionine. Now, MET is just the abbreviation for its name. I told you guys that I'm not asking you to memorize the names or the abbreviations. They're already gonna be on this page for you. But I do want you to know that they are amino acids. And when we take these amino acids and we link them all together, then we're building, finally here at the end, a whole protein. That's it. The whole thing is a protein when all of these blank lines are filled in and all the amino acids are linked together, and we start at a start and we finish at a stop. That's one protein. So let's take a look at how we find methionine and how we get to start, okay? So looking back at our chart right here, you can see that we've got 4 letter choices, and there's different columns for these letter choices. Our first letter right here is A. That's our first choice right there is A. So where is the first letter on this? Well, it's right here. That's our first letter, and we've got 4 options. We got a U, a C, an A, and a G. Well, we already know that our first letter is an A, right there. AUG, the first letter is an A. So what I'm gonna do is I'm gonna go to my first letter column, A. That means the amino acid I'm looking for is somewhere in that row right there. I'm not sure which one it is. It's one of these possibilities in here. But here's something else I know. I also know that it's not outside of this row. So it's none of these, and it's none of those. So my answer is right in this blue space somewhere. But I gotta narrow it down a little more. So what's my next letter? My next letter is a U. All right, so my next letter is a U, so I'm gonna go to my second letter column. There's my second letter column. And you can see I've got choices, a U, a C, an A, and a G. Well, I know my second letter is a U. So where does my U column cross with my A column? Right there. So what I've learned is that if I follow my A column across, my A column across and my U column down, they meet right there in that little space. So my answer is somewhere right here with these 4. And I also know that it's not anywhere there. So I've narrowed it down to just this box right here. Now I go to my third letter. My third letter is a G, right there. Okay, so where is the third letter? The third letter column is right here. And you can see we got lots of choices. There's some, there's some, there's some, there's some, but there's lots of G's here. There's a G, there's a G, there's a G there. Which G should I use? Well, I'm gonna use the G from the column I already know my answer is in. A told me that a long time ago, so that's what I'm gonna use. So how do I get there from this G? Well, what I have to do is I have to go backwards, right to left, which is kind of hard because we don't do that a whole lot. Right to left all the way over to my answer, which is right here. That's it, methionine. So this G right here tells me that my third letter answer is right there. I have to follow that over from right to left. So what that tells me is my amino acid is MET, which is already filled in, methionine. And then next to it, there's this word that says start. Okay, so these are the secret code words that we as Rockland teachers have added in here so that as you guys go through this, you'll be spelling different items. Let me give you an example. First letter we spell is a really short letter, it's 2 words. So my code, the codon I'm reading is UCG. Let's see if we can do that here. All right. U is my first column right here. So I know it's somewhere in that column. C is my second column, somewhere right there. So I know my answer. I know it's crossed out on here, but my answer is somewhere in that group right there. UC, and I'm gonna follow G back. So I go to my third letter, there's my G. Follow it all the way back, right there. And there's my answer. SER with the letter G. It's already filled in for us, so we can see that. Now the next one, it wanted us to look up the codon CAC. And CAC codes for HIS. HIS also matches up with a letter. Let's see which letter it matches up with. C, A, C. First row, first letter is C, second letter is A, third letter working backwards is C. And that matches up right there with histidine, which is the letter O. So what I do is histidine is already filled in. I'm just gonna put a letter O. And I spelled the word go. And if you keep going here, you're gonna continue to spell out some other kind of word. And if it doesn't seem like you're spelling something that makes sense, probably means that you need to go back and check somewhere on here that you're not transcripting DNA to RNA correctly or translating RNA to an amino acid correctly. So when you get all done and you take these amino acids and you add them all together, remember that those finally make a protein. And that's the whole big picture here is we're taking amino acids, the ribosome is sticking them together and making a protein. So we're actually doing the job of the ribosome right now. Thank you guys. If you have any questions, shoot me an email and I can help you out. Thanks a lot.